Hello everyone and welcome back to another CPD talk. So today we're going to be doing part 2 of the transfusion Titbits. I know it's been a little while since the last one. I did record the last one very close to the first one, but as I was recovering from COVID, it's full of me coughing, so we're going to go again. So this series is meant to be a short series with as much information in it as possible for people starting out in transfusion. Just to bear in mind that this particular presentation, because we're going through the basics of antibodies, is going to be a little bit longer because there's a lot to squeeze in. But hopefully it's still following a similar format. So last time we covered what blood transfusion actually is. Today we're going to cover some basics on antibodies. As many people probably know, there are 5 classes of antibodies that we produce, which are known as immunoglobulins. These include IgG, IgA, IgE, IgM, and IgD. In blood transfusion, as a rule, we do not look at all of these. We're only interested in the ones that will interact with red cell antigens, and this means for the most part, dependent on what you're doing in your blood transfusion career, we only look at IgG and IgM. So antibodies are typically found in patient plasma and are often produced as a result of an immune response. However, depending on the class of antibody, some will be naturally occurring in the body. They bind to antigens, mostly foreign ones, to indicate that these cells need to be destroyed. This happens with all antibodies, and quite often when you learn about antibodies, you're looking at it in terms of bacteria or viruses coming into the body. When we're talking in blood transfusion, it's following a similar principle, but we're looking at when we have foreign red blood cells entering the body. So we're going to walk through some of the basics of IgG and IgM class antibodies, specifically in relation to blood transfusion. I'm going to do that using this table. What we're going to do is compare and contrast each of these to do with IgG and IgM. As I said, those are the main antibodies that we're looking at in blood transfusion, so those are the ones I'm going to look at in the most detail. So we're going to start off with the structure. If you've looked at antibodies before, you know that the typical one that you'll look at is this sort of Y-shaped structure, which is the IgG antibody. It has 2 binding sites, and it's quite a small antibody. In comparison, IgM sort of looks like a snowflake, and it almost looks like it's made up of 5 units of IgG. If you were to look closer though, you'd be able to see that they're not exactly the same, and IgM is actually more similar to a different type of antibody. But I'm not going to spend time going through that. If that's something you're interested in, there are lots of resources online that you can look up to know about all 5 classes of antibodies. The main thing to take home from this is IgM is a lot bigger with a lot more binding sites than IgG. We're then going to look at reactivity. So when we're talking about reactivity, we're talking about when these antibodies will bind to an antigen. So IgG binds at 37 degrees or around that temperature, which is what we call body temperature. On the other hand, IgM will normally react at about 25 degrees, which again, that's what we call room temperature. There are some IgM antibodies that actually react even better at colder temperatures, so between 2 and 6 degrees. It depends on the specific antibody. Obviously, we're talking in generalizations here. We then want to look at the techniques that we would use in the lab to mimic what would happen in the body. So this is how we're actually detecting that there are these antibodies present. So IgG, we tend to use the indirect agglutination technique. IgM, normally we would use the direct agglutination technique, but it's important to note that it can also be detected by the indirect agglutination technique. Now, the reason that it's good to know about the different types of techniques are for the following. So if I start with the direct agglutination technique, IgM uses the DAT technique because it's a bigger antibody and it has 10 binding sites in total. To get it to react with antigens on red blood cells is a lot easier, therefore the agglutination occurs more readily. So we can literally just add antibody to an antigen and see a reaction. It also tends to be run, so the DAT tends to be done at room temperature, so we incubate it at 25 degrees. Conversely, the indirect agglutination technique, we need extra reagents to add to that to help with the binding for us to be able to visualize the IgG antibodies binding to antigens. It also, IAT is almost exclusively done at 37 degrees to mimic the body and to allow this reaction to occur. Finally, I want to talk about clinical significance. So clinical significance, or a clinically significant antibody, these are terms that you're going to hear a lot in blood transfusion. When we're talking about clinical significance, what we're looking at is how likely that antibody is to cause an immune response. So that immune response in blood transfusion, we're looking for either a transfusion reaction or a hemolytic disease of the fetus or newborn. So if we say an antibody is clinically significant, it means it's going to cause an immune response. Now, looking at all the information we have about IgG antibodies, the fact that they react at your body temperature, and that in order for us to detect them, we have to mimic body temperature, a good proportion of IgG antibodies are clinically significant. Not all of them, but a good proportion are, because the reactions occur just normally in your body. Conversely, IgM doesn't normally react at 37 degrees. Therefore, normally it's not clinically significant. So antibodies that are IgM are not normally clinically significant. The one big exception, because there's always an exception, is ABO. And that's because it uses a different pathway to be clinically significant. And ABO incompatibilities are normally the most deadly. So it's sort of saying IgM isn't clinically significant, apart from in this case where it's the most dangerous. And I have a talk about acute hemolytic transfusion reactions if you want to know more about that in more detail. Right. So how does this relate to blood transfusion testing? I've kind of touched on this, but let's go into it in more detail. So we perform two main routine tests, and these are blood grouping, where a patient's ABO group is determined, and antibody screening, where we look for any other antibodies which could cause an adverse reaction to a transfusion. When we test for a patient's blood group, we start by performing a forward and reverse group. The forward group will look at what antigens the patient has present on their red blood cells by using reagent antisera with known ABO antibodies in. And I will tell you right now, when you're first starting out in blood transfusion, getting the antigen versus antibody understanding down can take a little while, but just remember that antigens are on the red blood cells, the antibodies are in the patient plasma. So when we're saying that the forward group is looking at the antigens that the patient has, so we're looking at the red blood cells, whereas the antisera, we're basically using a reagent plasma with known antibodies in it. The reverse group is then used to confirm that this is an accurate result by looking at what antibodies the patient has in their plasma against known reagent red cells with known antigens on them. So you can see here I have an example. A lot of blood grouping is done in these cards, if you haven't seen them before. The red arrows that I have are looking at the forward group, so you can see which antigens are present on the patient's red blood cells. When it comes to reading these cards, we're looking at whether or not red blood cells have stuck to an antibody. And these little cards kind of work like a filter in a way. So if you have a reaction, then you will expect that the cells will be towards the top of the card. And if there's no reaction, then the cells will still be separate so they can get to the bottom of the card, if that makes sense. So if we're to look at the forward group here, you can see that there's a line at the top of the card for the A and a line at the top of the card for the D. Therefore, they have a reaction in the A and a reaction in the RHD. So the forward group would indicate that they are an ARHD positive patient. Then all of our tests will have, well, the majority of our tests will have a control within it, just to make sure that we're not getting inaccurate results. The control should always be negative. If it's positive, then we chuck out the result. So here, all of the red cells are at the bottom. That indicates that the control is negative. And then on the right-hand side of the screen, you can see we've got the blue arrows. So this is looking at the reverse group. The cells here have dropped to the bottom of the A, but they're at the top of the B. So if you've got a patient who is a group A positive patient, you would expect them not to have the A antibody, because if they had the antibody to their blood group on the red cell antigens, then they would be attacking their own cells and they would be very unwell. So what we expect them to have is the opposite. So this is a group A patient, so they should have anti-B. And if you have a look at this, they're reacting with the B, so they have the B antibody. Therefore, this patient has presented as a group A RHD positive patient. So when we're determining a patient's blood group, the antibodies that we look for and the reagent antisera are IgM. And because of this, the testing is normally performed at room temperature. So we don't incubate at 37 degrees, we incubate at 25 degrees. And it uses the direct agglutination technique. On the other hand, antibody screening, which is what we do alongside the ABO grouping, is looking for clinically significant antibodies other than ABO. This involves a 3-cell screen using reagent red cells with a variety of known antigens present to which the patient's plasma is added. General idea of this screen is that if it is negative, then the patient doesn't have any other antibodies which are commonly clinically significant. However, if it's positive, then we will need to investigate further to determine what antibody or antibodies that they have present. So this is then predominantly looking for IgG antibodies. So therefore, the test is incubated at 37 degrees to mimic the body temperature, and the indirect agglutination technique is used. Because we're looking for that different type of antibody, we're changing the technique. And that's why it's so important to understand what we're actually looking for in terms of the type of antibodies present and how that impacts our testing, because then when our testing isn't normal, we understand what could be causing it. And that's the basic of antibodies relating to blood transfusion. Next time we're going to be covering routine grouping results, which should help understanding these interactions a bit more, because I kind of went through it quite quickly, as well as how we determine what blood group patients are. Hopefully this has been a useful introduction into antibodies. There are lots of great resources out there if you are interested in knowing more. So thank you very much for listening. Please remember to do reflective learning on any CPD that you undertake to make it robust. And I look forward to the next one. Bye!